Yes, good afternoon to all. Right. What's the last topic? Okay, right. So next we'd like to start the biotechnology part. So next, biotechnology. So biotechnology. So it is an applied branch of science in which various types of living organisms are used to produce various valuable products, okay, by using sophisticated techniques or equipment that is called as biotechnology. So it is a branch of science, it is an applied branch in which the living organisms like plants, animals, the microorganisms can be utilized to produce the valuable products by using sophisticated techniques or equipments that is called as a biotechnology. Okay, actually the term biotechnology is new one, but the process involved in this biotechnology is as old as human civilization. So based on the origin of this biotechnology or growth of the biotechnology, it have been divided into two types. One is traditional biotechnology, and other one is, yes, completed. Okay, right, we'll continue that one. Yeah. Okay, traditional biotechnology, modern biotechnology. Traditional biotechnology means the biotechnology which is based on the natural capabilities of the organism, natural characteristic features of the organism. And the modern biotechnology means which is based on the DNA composition, DNA structure, and also it is based on rDNA technology. This is a modern biotechnology. Okay, right. Then the biotechnology has vast scope. So this biotechnology can be applied in different fields. So based on that, the biotechnology has given different names. So these includes, you remember the red biotechnology, green biotechnology, white biotechnology, blue biotechnology. So then bioinformatics. So based on the applications of this biotechnology in different fields, it has given different names. So red biotechnology means the application of biotechnology in medicine or in healthcare sector. The application of biotechnology in medicine or in healthcare, that is red biotechnology. Green biotechnology, application of biotechnology in agriculture. Just to remember these terminologies. Application of biotechnology in industry or industrial application of biotechnology is called as white biotechnology. Blue biotechnology, application of biotechnology for the improvement of marine flora and fauna. For the improvement of marine flora and fauna. Here flora means plants, fauna means animals. So applications of biotechnology for the improvement of marine or aquatic organisms. So bioinformatics, because simply you can say biotechnology plus information technology. That is also called as computational biology. That means the various software techniques can be applied. So say for example proteomics, genomics is a part of bioinformatics. Proteomics and genomics, the protein structures can be assessed, genome structures can be assessed by using softwares, by using algorithms, that we call as bioinformatics. So simply it is a mix of biotechnology and information technology. So these are various branches of biotechnology based on its application. So simply you remember this terminology. Red biotechnology means application of biotechnology in medicine. Green biotechnology in agriculture. White, industry. Blue, improvement of marine flora and fauna, that is aquatic. So then bioinformatics, biotechnology plus information technology. Got it? Right. Suppose if you observe this biotechnology in medicine, the biotechnology has vast applications, mainly medicine or agriculture, whatever it may be. Here, the biotechnology can be applied in the production of various types of vaccines. The biotechnology is applied in the production of vaccines, monoclonal antibodies, interferons, in cloning process, stem cell therapy, gene therapy, gene editing, So three parent baby, IVF, in vitro fertilization, DNA fingerprinting, These are some important applications of biotechnology in medicine. In agriculture, we are producing GM crops. You have to remember this one, GM crops, genetically modified crops, or you can say transgenic crops. And we are using biofertilizers in agriculture, biopesticides, biofuels, and also in the environmental cleanup process, we are using bioremediation process. In industry, we are producing various types of therapeutic proteins, and various types of enzymes. So these are produced in large scale in industry. So these are all the applications of biotechnology. If you complete this one, the biotechnology will be over. Okay. So we have to study these topics in biotechnology. So vaccines. Nowadays we are producing various types of modern vaccines by using rDNA technology process. Okay. Monoclonal antibodies, MAPS, interferons, cloning process, stem cell therapy, gene editing process. So three parent baby concept. We have developed three parent baby concept. Then IVF technology, in vitro fertilization, the DNA fingerprinting or DNA testing or DNA analysis. Then in agriculture, we are producing GM crops, genetically modified crops or transgenic crops. We are using biofertilizers, biopesticides or biofuels. Then in an environmental cleanup process also, we are using biotechnology that is called as bioremediation process. And also industrially, we are producing therapeutic proteins in large amounts or various types of enzymes. Okay, these are the various applications of biotechnology in different fields. So let us see one by one. Over. Right. Let us see the vaccines. So what are vaccines? So what is meant by vaccine? Any preparation which is used to trigger our immune system. Okay, any preparation which is used to trigger our immune system or which can enhance, which can boost our immune system, that is called as a vaccine. So generally the vaccines are of two types. So one is traditional vaccines, vaccines and modern vaccines, or these are also called as recombinant vaccines. Recombinant vaccines. So what are traditional vaccines? What are traditional vaccines? The vaccines which are produced by the traditional methods are crude methods. Crude methods. That means in traditional vaccines, a complete pathogen, the whole or the complete pathogen can be used. The complete pathogen is used in the preparation of the vaccine. Generally, the vaccine, in vaccine preparation, the pathogens which are responsible for the diseases can be inactivated or they can be heat killed, and those can be used as vaccines. The complete pathogen is used here. Okay, so for example, attenuated vaccines, attenuated vaccines, then inactivated, this is also called as killed vaccines. Attenuated, or it is also called as live vaccines, killed vaccines. Then toxoids. These are different types of traditional vaccines. Attenuated or live vaccines. That means the pathogen is weakened. In attenuated vaccine, here the pathogen is weakened. It is not completely killed. The pathogen is weakened by physical methods, by physical methods. That means when it is exposed to UV light, or high temperature, then the pathogen may be weakened. So that means when the pathogen is exposed to UV light or high temperature for a short period of time, then the vigor of this pathogen will be reduced. Okay, the potentiality, that means the vigor of this, that means virulency means disease causing capacity of the pathogen will be suppressed, decreased. And these type of vaccines are called as live vaccines. So live vaccines mean the vaccine is not completely, that means the pathogen is not completely killed, simply it is weakened. And these type of vaccines are by chemical methods. That means by using chloroform, some chemicals like by using chloroform, phenols, alcohols, etc. By using these chemicals, we can weaken the pathogens. So these are called as attenuated or live vaccines. And inactivated or killed vaccines. Inactivated or killed vaccines means the pathogen is completely killed. The pathogen is completely killed. The pathogen is completely killed. Means what? When the pathogen is exposed for a long period to these UV radiations or temperature or to these chemicals, the pathogen will be killed. So these type of vaccines are called as inactivated or killed vaccine. Suppose say, for example, OPV vaccine. Example of this attenuated vaccine is OPV. OPV means oral polio vaccine. This OPV is an attenuated vaccine or live vaccine. Here the pathogen, the poliomyelitis virus, is not completely killed, simply it is weakened by adding some chemicals or by exposing to UV radiations or high temperature. These are called as attenuated or live vaccines. And then inactivated or killed. Inactivated or killed means it is the pathogen is completely killed. When you expose this pathogen for a long period to these chemicals, for a long period to this UV radiation, temperature, then the pathogen will be killed. Then this is called as inactivated or killed vaccine. For example, pertussis. Pertussis vaccine. DPT vaccine is there, DPT. diphtheria, pertussis, and tetanus. In that, pertussis is killed vaccine. Here, the Bordetella pertussis bacteria is completely killed. Okay, and then toxoid. Right. Here, then toxoid. So toxoids means toxoid. Suppose say the pathogen is there. Suppose say this is a bacteria. So this is a bacteria, and this bacteria releases certain chemicals, or you can say toxins. Toxins. That means the part of the bacteria or the toxins released by the pathogen. These toxins are extracted, and these toxins are purified, and these are added with some adjuvants. Adjuvants means some chemicals can be added, or some preservatives can be added. So the toxoids means what? The toxins released from the pathogen are extracted, and they are purified. They are mixed with some other substances called adjuvants, and these adjuvants acts as preservatives. Is it clear? And these type of these type of vaccines we call as toxoids. Say for example, DT, diphtheria toxoid, TT, tetanus toxoid. These are called as toxoids. Toxoids means the toxins released by the pathogens are purified, and it can be used as a vaccine. These are all called traditional vaccines. Okay. Say, let us see some important examples. So, examples of all these traditional vaccines. OPV, oral polio vaccine. TPT, it is diphtheria, pertussis, and tetanus. So this is called as triple antigen or triple vaccine. So DPT vaccine, it is against 3 diseases: diphtheria, pertussis, and tetanus. Then BCG, you remember the abbreviation Bacillus calmette and gurin. This is against TB, BCG. TB, and also this DT, diphtheria toxoid, and TT, tetanus toxoid. These are all the examples of traditional vaccines. Okay, and also measles vaccine. Rabies vaccine. Over. Right. So next, let us see modern vaccines. Modern or recombinant vaccines. Modern or recombinant vaccines. Here, the vaccines, these vaccines can be prepared by, these vaccines can be prepared by rDNA technology. rDNA technology process, recombinant DNA technology process, or by genetic engineering process. Genetic engineering. These vaccines can be prepared by using rDNA technology or by genetic engineering process. So that means in traditional vaccines, in the preparation of traditional vaccine, we use the complete pathogen. We use the whole pathogen. But here we are not using the whole pathogen. A part of the pathogen which is responsible for the disease, that can be used. For example, hepatitis B vaccine. Hepatitis B vaccine. Hepatitis B vaccine. Suppose say this is hepatitis B virus, and it contains the genetic material. Okay, and this hepatitis B virus, it releases a protein. This is called as HPSAG. HPSAG. HPSAG means hepatitis B surface antigen. Hepatitis B surface antigen. So actually, the gene responsible for this one, the gene, so the DNA responsible for this hepatitis B, that is HBsAg, it can be extracted. Okay, and it can be cloned. Okay, thereby will construct a rDNA, recombinant DNA. So that means here we are not using the complete pathogen here. The gene or the DNA responsible for this hepatitis HBsAg protein, simply this DNA is extracted. Are you getting? So the DNA is extracted. Now this DNA is inserted into another cloning vector, that is another DNA. Thereby will construct a rDNA, recombinant DNA. What is meant by recombinant DNA? Recombinant DNA means the DNA formed by the construction of two or more different types of DNAs. So this is one DNA, this is another DNA. These two DNAs are combined together. Thereby will form a new DNA. That new DNA is called as rDNA, recombinant DNA. Okay. So no need to go in depth of this process. Just you remember the terminology. Recombinant DNA. This rDNA is constructed. Now this rDNA is inserted into the host organism. Now the host is fermented. That means will provide the food material to this host. This bacteria or yeast or host will multiply. So thereby the rDNA also multiplies. So multiple copies of the DNA can be produced. From this, the desired protein can be extracted. That is HPSAG protein. So that means simply you understand, here we are not using the whole pathogen. Simply we are using the gene. Simply we are using the protein which is responsible for the disease. This HPSAG protein is responsible for the disease. And this protein is produced in large amounts. Is it clear? So these type of vaccines we called as modern vaccines or recombinant vaccines. That means in modern vaccine preparation, we are not using the complete pathogen. Simply we are using the DNA responsible for the protein. Is it clear? So a part of the DNA is extracted and this DNA is inserted into the cloning vector. Thereby will construct a rDNA. Now this rDNA is introduced into the host organism. So this host organism is multiplied. From this, multiple copies of the protein can be produced. And this protein is extracted, it is purified, it can be used as injection in the form of it can be used as vaccine in the form of injections. Is it clear? So this type of vaccines are called as modern vaccines or recombinant vaccines. So simply you remember, by this biotechnology process, we can able to produce various types of traditional and modern vaccines. Traditional and modern vaccines. Clear them? Alright. And next one is monoclonal antibodies. Then monoclonal antibodies. Monoclonal antibodies. These can be represented with maps. Monoclonal antibodies. So what are monoclonal antibodies? Generally, listen. Generally, the antibodies produced in your body are polyclonal antibodies. First of all, what is meant by antibody? Antibodies are the protective proteins which are released by our immune system to fight against the antigens. Antigen means any foreign particle enters into our body which causes harm, then it is called as antigen. So against this antigen, some proteins are produced in our body. These protective proteins are called as antibodies. Generally, the antibodies released in our body, they are polyclonal type. Polyclonal type means, suppose say this is a one polypeptide chain of antibody, this is another polypeptide, another like this. So many polypeptide chains are there. These polypeptide chains are formed from different cells. So that's why it is called polyclonal. But here, what is monoclonal? Name itself indicating mono means single. Clone means what? Group. Clone means group. So this is a single group of cells. All cells are similar here. So from these cells, same type of antibodies are formed here. That means monoclonal antibodies means homogeneous preparation of antibody molecules. Monoclonal antibodies means homogeneous preparation of antibodies from a single clone of cells. So homogeneous preparation of antibody molecules, homogeneous preparation of antibody molecules from a single clone of cells. From a single clone of cells called as homogeneous preparation of antibody molecules from a single clone of cells. From a single clone of cells called as monoclonal antibodies. Monoclonal antibodies. So these monoclonal antibodies are used in the diagnosis. They are used in the diagnosis of various diseases. In the diagnosis of various diseases, and also they are used in the treatment of various diseases. These monoclonal antibodies are represented by MAPs, monoclonal antibodies. So these monoclonal antibodies are used in the diagnosis and treatment of various types of diseases. Various types of diseases, including some types of cancers also. Including some type of cancers. And these monoclonal antibodies are produced by a process called as hybridoma technology. You remember this terminology? Hybridoma technology. Hybridoma technology. The process used in the preparation of these monoclonal antibodies is hybridoma technology. So what is hybridoma technology? In this hybridoma technology, suppose say this is B cell. B cell means what? B lymphocyte cell. The B lymphocyte cell can able to produce the antibodies. Okay. And this is a myeloma cell. B lymphocyte cell, and say this is myeloma cell. Myeloma cell means it is called cancer cell. Cancer cell. Here the literal meaning of cancer is multiplication. Uncontrolled multiplication is called as cancer. Uncontrolled growth is called as cancer. Here the B cell is combined with a myeloma cell, thereby we'll get a new cell. That new cell is called as a hybridoma cell. Hybridoma cell. So now listen. So this B cell is having antibody producing capacity, and myeloma cell, it is having multiplication capacity. Okay, it is undergoing multiplication continuously. So when these two cells are combined together, we will get a hybridoma cell. Now this hybridoma cell is having two properties. One is antibody producing capacity, another one is multiplication capacity. Okay. Now this hybridoma cell, it is grown in the laboratory. Say this is hybridoma cell. It is grown in the laboratory. As it is having multiplication capacity, from a single hybridoma cell, many hybridoma cells are formed. And from these hybridoma cells, same type of antibody is released. Similar type of antibodies are released. And these antibodies are called monoclonal antibodies. These antibodies are called monoclonal antibodies. Suppose if you want to raise antibodies against hepatitis, if you want to produce antibodies against hepatitis disease, simply you can infect this B cell with hepatitis B virus. Are you getting? When you infect this B cell with the hepatitis B virus in the laboratory, the B cell will be sensitized against this hepatitis B. So the B cell can release the antibodies against hepatitis B. In a similar manner, you can raise the antibodies against any type of disease. And these type of antibodies you call as monoclonal antibodies. Monoclonal antibodies. Are you following or not? Following na? Okay, right. So simply you remember, in exam point of view, you need what are monoclonal antibodies. And what method is involved in the production of monoclonal antibodies. This you have to remember, that's enough. So monoclonal antibodies means homogeneous preparation of antibody molecules from a single clone of cells, group of cells. From all group of cells, same type of antibodies are released. These are called monoclonal antibodies. And they are used in the diagnosis, treatment of various diseases, including some cancers. Then these monoclonal antibodies can be produced by hybridoma technology. What is hybridoma technology? The B lymphocytes are combined with the myeloma cell to form a hybridoma cell. Now these hybridoma cells are grown in the laboratory. They are multiplied. These hybridoma cells releases the antibodies which are all similar. And these type of antibodies you call as monoclonal antibodies. Monoclonal antibodies. Got it? All right. And the next one is interferons. 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 So these interferons are the proteins released from the protective proteins. Protective proteins released from the virus infected cells. Released from the virus infected cells. Virus infected cells or cancerous cell. Suppose say this is cancer cell. That means when the cell is infected by oncovirus. What is oncovirus? What is oncovirus? It is a cancer causing virus. Oncovirus means cancer causing virus. When this virus infected a normal cell, it becomes cancerous. From this cancerous cell, certain proteins are released. Okay, these proteins are called interferons. So simply you remember the definition of these interferons. The interferons are the proteins released from the virus infected cell or cancerous cell, which are protective in nature. And they are used in the treatment of cancers. They are used in the treatment of cancer, various types of cancers. Okay, there are various types of interferons. Alpha interferons, beta interferons, gamma interferons. These are different kinds of interferons are there. They are used in the treatment of various types of cancers like breast cancer, lung cancer, blood cancer. Over. Over. So simply you remember interference. What are interferons? These are the proteins released from the virus infected cell. Virus infected cell means cancer cell. Cancer cell. So this oncovirus, you remember this terminology. Oncovirus means cancer causing virus. When this oncovirus infects a normal cell, it becomes cancerous. From this cancerous cell, certain proteins are released. Okay, these proteins are called interferons. And these interferons will protect the neighboring proteins, neighboring cells. The interferons will protect the neighboring cells from getting cancer. Okay, to some extent, these interferons protects from cancer cell. And these type of interferons can be produced in large scale by using rDNA technology process. Now these interferons are used in the treatment of cancer. There are various categories of interferons, alpha, beta, gamma interferons are there. Okay, right. Next, cloning. 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 So what is cloning? The production of living structures or organisms. Production of living structures or organisms which are genetically identical to their parents, which are genetically identical to their parents. So, what is cloning? The production of living structures. Living structures means that may be cell or that may be a genetic material that is part of DNA. So, the production of living structures are complete whole organism which are genetically identical to their parents. That is called as a cloning process. Okay. Here the literal meaning of clone is branch. The literal meaning of clone is branch, or you can say fig. So cloning is the production of living structures or organisms which are genetically identical to their parents. These are the Xerox copies to their parents, you can say. Okay, right. And this cloning is of different types. One is gene cloning, cell cloning or cellular cloning, and organism cloning. Cloning is of different types. Gene cloning, cellular cloning, and organism cloning. Gene cloning, cellular cloning, and organism cloning. So what is gene cloning? Suppose say this is our desired DNA. Say this is our desired DNA. Okay. If this DNA is copied, if this DNA is multiplied, a large amount of DNA is produced, then this DNA, this cloning you call it gene cloning. That means a part of the genetic material, a part of the DNA is multiplied. So what is the process involved here? Example, by rDNA technology process, by recombinant DNA technology process, or by PCR method. PCR means what? Polymerase chain reaction. By PCR process, by PCR method, by polymerase chain reaction, or by rDNA technology, we can able to multiply the DNA, required DNA, desired DNA. Suppose say for example, insulin. Do you heard about insulin? What is insulin? Insulin is a hormone which is used to treat diabetes, type 1 diabetes. Now the insulin injections are available. How this insulin injection is produced? By rDNA technology process, by recombinant DNA technology process. Actually, the insulin protein is released in the pancreas, released by the beta cells of the pancreas. So, in some persons, the insulin production is decreases. Thereby it leads to diabetes. So in these patients, we are injecting the insulin protein. That insulin protein is produced by this rDNA technology process. That means simply the insulin gene is extracted, the insulin DNA is extracted. Now that insulin DNA is cloned with a plasmid DNA. That is, thereby recombinant rDNA is constructed. Now the multiple copies of the rDNA, the multiple copies of the insulin protein is produced, and from this the insulin protein is extracted. Insulin protein is extracted. Now this insulin protein is purified, and now we are using it as an injection to treat diabetes. So like that, by cloning process, we have produced the insulin protein in large amounts. That process is called as gene cloning. Gene cloning. Is it clear? And next, another one is cellular cloning. Just now we discussed about this what we called monoclonal antibodies. The monoclonal antibodies are produced by hybridoma technology. So that means cellular cloning. Here in cellular cloning, many hybridomas are produced like this. So this is example of hybridoma technology. The production of many hybridomas. It is an example of cellular cloning. Okay. So cell cloning means from a single cell, multiple copies of the cells are produced. This is called cellular cloning. Right? Example of this cellular cloning is hybridoma technology. By this hybridoma, we can able to produce the similar type of cells. And next, let us see organism cloning. So organism cloning. Remember? Organism cloning. This was given in previous exam. So organism cloning means if an entire organism, a whole organism is produced from a single parent, which is genetically identical. If the whole organism, suppose say for example, you take any hair follicle. Suppose you take a hair follicle. From this hair follicle, you can extract the DNA molecule. By using the DNA, you can produce the whole structure, whole organism. Or you can take the DNA from a tissue, DNA from a skin cell, DNA from any part of our body. The DNA is enough to produce the entire organism. That we call as organism cloning. Here, the human cloning is banned here. Just you remember, organism cloning means production of or the formation of organism cloning means the formation of one or more genetically identical individuals, from a single parent. So this organism cloning is an example of reproductive cloning. So this organism cloning is an example of reproductive cloning. So the formation of one or more individuals which are genetically identical, which can be produced from a single parent. And here this organism cloning is an example of reproductive cloning. The question was given in the previous UPSC prelims exam. So the biotechnological process involved in the reproductive cloning process is, he has given various options. And the option here is the reproductive cloning. The method involved in the reproductive cloning is SCNT. SCNT means what? Somatic cell nuclear transfer. This was given. The process involved in the reproductive cloning process is, SCNT, somatic cell nuclear transfer technology. SCNT, somatic cell nuclear transfer technology. So what is reproductive cloning? if the entire whole organism is produced from a single parent, which is genetically identical, then it is called reproductive cloning. The process involved in this reproductive cloning is SCNT, somatic cell nuclear transfer technology. Okay. So example of this reproductive cloning, dolly. The formation of dolly by a scientist called Ian Wilmot. Ian Wilmot. So production of dolly by Ian Wilmot. What is dolly? It is a cloned sheep. Dolly, it is a cloned sheep. Dolly is a cloned sheep. So this dolly is the first cloned mammal in the world. First cloned mammal. Cloned sheep or first cloned mammal. First cloned mammal. Right. Just for your understanding, I'll let you know what is somatic cell nuclear transfer technology. Okay, in exam point of view, in preliminary point of view, just you have to remember the process involved in the reproductive cloning process or organism cloning process is somatic cell nuclear transfer technology. Okay, so the best example is the production of dolly by Ian Wilmot. Let us see how this dolly can be produced. It is for your understanding only. What is somatic cell nuclear transfer? So first, he has taken a sheep called Finn Dorset ewe. Ian Wilmot has taken a sheep called Finn Dorset ewe. It is a female sheep. Female sheep. He has collected udder cell. Udder cell. Udder cell means it is a vegetative cell. Udder cell. It is a vegetative cell. He extracted the nucleus. He isolated the nucleus of this udder cell. See, this is Finn Dorset ewe. From this Finn Dorset ewe, he has taken udder cell. Udder cell means it is a vegetative cell. From this vegetative cell, he isolated the nucleus. Okay, he separated the nucleus. Now, he has taken another sheep called Scottish blockface. Scottish blockface. Okay, now he collected the egg cell. Egg cell. He removed this egg cell. He removed the nucleus of this egg cell. He removed the nucleus of this egg cell. He has taken the empty egg cell here. Okay. Now, he inserted the nucleus of this Finn Dorset ewe into the empty egg cell of the Scottish blockface. Got it? He has taken the vegetative cell called udder cell. From this vegetative cell, he has taken the nucleus. He separated the nucleus. Now he inserted this nucleus into the empty egg cell of the Scottish blockface. So this is Scottish blockface is also female sheep. This is also female sheep. Got it? Now, in the next step, he has taken another Scottish blockface. Another Scottish blockface. This is also female sheep. This is also female sheep. He inserted this egg cell into the womb of this second Scottish blockface. Okay. After completion of gestation period, this second Scottish blockface, which is acting as a surrogate mother, has given birth to a sheep called Dolly. And this Dolly is almost similar to Finn Dorset ewe. It is a Xerox copies of Finn Dorset ewe. It is not having the characteristic features of the Scottish block phase. It is a clone of Pindorcetu because the genetic material, the nucleus is from Pindorcetu. So it is considering as a clone of Pindorcetu. Here there is no involvement of male sheep, only female sheep are involved. Got it? Okay. So we can say that this dolly is the Xerox copy of Pindorcetu. Like that, the whole organism can be produced by this cloning process. And this is called as organism cloning. This is called organism cloning. And the process involved here is called as somatic cell nuclear transfer. Why it is called somatic cell? Because udder cell is a somatic cell, that is vegetative cell. Somatic means vegetative. The vegetative cell, so the nucleus of this vegetative cell is transferred. Somatic cell nuclear transfer. Okay, this process is involved in the reproductive cloning. Got it? Have any doubts? Yes. Here there is no involvement of male sheep here. There is no sexual reproduction process here. Simply the genetic transfer of the material. Okay, genetically the material is transferred here. So this process is called somatic cell nuclear transfer technology. This was given in the previous exam. Is it clear? Right. This is a cloning process. Let us see animal cloning in India. Animal cloning in India. So various animals have been cloned in India. Samrupa, Garima, Mahima, Shrestha, Swaran, Nuri, Purnima, Hisar Gaurav, Sarb Gaurav. These are all cloned animals. So write down animal cloning in India. Animal cloning in India. Samrupa. 2009. First cloned buffalo. First cloned water buffalo. Samrupa, first cloned water buffalo. Cloned at NDRI, Karnal, National Dairy Research Institute. Samrupa, 2009, first cloned water buffalo. First cloned water buffalo, cloned at Karnal, NDRI Karnal, National Dairy Research Institute. National Dairy Research Institute. Next, Garima 2. Garima 2. Garima 2. It was cloned in 2010. Garima 2. It is also cloned buffalo. Cloned at NDRI Karnal. Cloned at NDRI Karnal. Next, Shrestha. Shrestha, 2010. First cloned male buffalo. Shrestha, first cloned male buffalo. First cloned male buffalo. Cloned at NDRI, Karnal. Next, Swaran. Swaran, second cloned male buffalo. Second cloned male buffalo. Cloned at NDRI, Karnal, 2013. Next, Mahima. Mahima 2013. The cloned buffalo Garima 2. The cloned buffalo Garima 2 gave birth to a calf, CLF, calf called Mahima. The cloned buffalo Garima 2 gave birth to a calf, CLF, calf called Mahima at NDRI Karnal. Next, Noori. The Pashmina goat. Cloned Pashmina goat. The first cloned Pashmina goat at Sher-e-Kashmir University of Sher-e-Kashmir University of Agricultural Sciences and Technology. At Sher-e-Kashmir University of Agricultural Sciences and Technology. So Pashmina goat, it is the first cloned goat. That is Noori. Noori, it is a first cloned Pashmina goat. And next, Hisar Gaurav. Hisar Gaurav 2015. Hisar Gaurav 2015. The cloned buffalo developed by the scientists of ICAR SERB. Scientists of ICAR SERB. ICAR means Indian Council of Agricultural Research. SERB means Central Institute of Research on Buffaloes. SERB, Central Institute for Research on Buffaloes. Next, Sarb Gaurav, another one. Sarb Gaurav. (2016). Sarb Gaurav 2016. An offspring of cloned buffalo. An offspring of cloned buffalo produced by SERB. That SERB Gaurav is an offspring of cloned buffalo produced by SERB. Right? So these are some cloned animals in India. So next, put subheading Lacons. Lacons means laboratory for conservation of. Laboratory for conservation of endangered species. Laboratory for conservation of endangered species. So Lacons means it is a laboratory for the conservation of endangered species. So what are endangered species? Endangered species means threatened species. So those species which are facing a high risk of threat from human activity, that is called as endangered species. the species which are facing a high risk of threat from human activity, that is called as a endangered species. Okay, so laboratory for conservation of endangered species, in order to support both in situ and ex situ conservation of various species which are facing a risk, which are threatened from human activities. A laboratory have been established in the year 1998. So this LACONS was established in an innovative manner to support both ex situ and in situ conservation of biodiversity. So it was established in the year 1998. It was supported by DBT, CZA and CSIR. LACONS was established in assistance from DBT. DBT means Department of Biotechnology. Department of Biotechnology. CZA, Central Zoo Authority. Central Zoo Authority. And CSIR, Council of Scientific and Industrial Research. Council of Scientific and Industrial Research, CSIR. CSIR. LACONS means Laboratory for Conservation of Endangered Species. It was established in the year 1998 in order to support for both ex situ and in situ conservation of endangered species. It was established with support from DBT, Department of Biotechnology, and CZA, Central Zoo Authority, and CSIR, Council of Scientific and Industrial Research. Council of Scientific and Industrial Research. Over? Right. Next, IVF, in vitro fertilization. Next, IVF. In vitro fertilization. First of all, what is meant by fertilization? Here fertilization means, what is fertilization? Union of male and female gametes. The union of male and female gametes is called as fertilization. Naturally, in humans, the fertilization occurs in the fallopian tubes. So, if you observe the reproductive system of females, say this is reproductive system of females, this is ovary, this is fallopian tube, and this is uterus, right? So generally the fertilization takes place in the fallopian tubes. Here the fertilized egg is formed. Okay, so the fertilization occurs in the fallopian tube, but some persons, some couples are suffering with conceiving problems. So that means in males sometimes the production of sperms will be reduced due to hormonal imbalance or due to varicose veins or whatever may be the reason. The production of sperms decreases in males, whereas in females due to hormonal imbalance or due to PCOD problems, polycystic ovarian disorders, the menstrual cycle become irregular. So the ovum or egg will not be released on time. So in this condition, the pregnancy will be delayed, the conceiving will be delayed. So in order to overcome all these infertility problems, conceiving problems, so We are following this IVF technology. In vitro fertilization. In vitro, what is meant by in vitro? In vitro means in the laboratory. In vitro means in the laboratory. Fertilization means union of male and female gametes. Here in this in vitro fertilization process, a healthy sperm is collected from the father and ovum is collected from the mother, and these two get fertilized in the test tube in the laboratory. These two get fertilized in the test tube in the laboratory. In vitro fertilization, it is a complex series of procedures. It is a complex series of procedures used to help with fertility or prevent genetic problems. It is a complex series of procedures. Used to help with fertility or prevent genetic problems. And assist with the conception of a child. This is called IVF technology. So, in this IVF technology, in vitro fertilization, a healthy sperm is collected and a ovum is collected. These two get fertilized in the laboratory in test tube. Okay. So, after some time, the embryo will be inserted into surrogate mother, or the embryo can be inserted into the mother's womb. If the original mother's womb is not suitable, okay, for pregnancy, then we will go for surrogacy. So this is called surrogacy. The baby delivered, we call it as test tube babies. And the baby delivered, we call it as test tube babies. So this is called as in vitro fertilization. What is in vitro fertilization? So it is a series of procedures in order to prevent the genetic abnormalities or conceiving problems, fertility problems, in which the sperm and ovum are collected and they are fertilized in the laboratory separately. In vitro means in the laboratory. In vivo means within natural conditions. So this fertilization process can be done in vitro, that is in the laboratory, and the babies born are called as test tube babies. The first test tube baby in the world was Louis Brown. She was born in the year 1978. The first test tube baby in the world was Louis Brown. So this IVF technology is the most effective form of assisted reproductive technology. So it is the most effective assisted reproductive technology. IVF technology is the most effective assisted reproductive technology. Over? Right. And the next one is 3-parent baby. And next one is 3-parent baby concept. Before going to this 3-parent baby, so generally, you know very well about this mitochondria. You heard about mitochondria. So actually, this is mitochondria. Say this is mitochondria. Actually, this mitochondria is called as a powerhouse of the cell. The mitochondria is called powerhouse of the cell. So in this mitochondria, a small amount of DNA is present. The circular DNA is present. This is called mitochondrial DNA. Or you can say mitochondrial genome. Got it? That means the mitochondria is having a separate DNA. That's why it is called semi-autonomous cell organelle. Okay. And here, this mitochondrial DNA inheritance is maternal. Inheritance is maternal. What means what? It is inherited to the next generation from the mother. That means the transmission, the inheritance of this mitochondrial DNA occurs from the mother, not from father. Got it? So, the mitochondrial genome, the mitochondrial DNA inheritance is from mother, not from the father. Is it clear? Right. Suppose if this mitochondrial DNA is abnormal in the mother, this abnormal mitochondrial DNA will be inherited to the next generation, to the babies. Are you getting my point? If the mother's mitochondrial DNA, because it is inherited from the mother, it is maternal. If the mother's mitochondrial DNA is abnormal, then this abnormal DNA will be inherited to the children. When it is inherited to the children may suffer with some metabolic abnormalities. The children may suffer with some metabolic disorders, long-term disorders. Is it clear? In order to rectify these metabolic disorders, in order to rectify these mitochondrial abnormalities, that is, the scientists have brought a concept called three-parent baby concept. In this three-parent baby concept, the abnormal mitochondrial DNA of the mother is replaced with the donor's mother DNA. Are you getting? So the abnormal mitochondrial DNA of the original mother is replaced with the donor's mitochondrial DNA. Okay. Then this concept we call as three-parent baby concept. That means what? So in three-parent baby concept, so the father DNA is inherited. That means father, from the father, same 23 chromosomes. 23 chromosomes. And then from mother also 23 chromosomes, normal. And from donor mother is having mitochondrial DNA. Got it? So that means the baby contains, suppose say this is a cell and this is a nucleus. This here 46 chromosomes are there. That is 23 chromosomes from father, 23 chromosomes from mother. Another DNA is there. This is donor's mother. Mitochondrial DNA. Mitochondrial DNA from donor mother. That means the baby is having three different DNAs. One from original father, one from original mother, another from donor mother. That is mitochondrial DNA from donor mother. Got it? And this type of a concept we call as three-parent baby concept. What is three-parent baby? So in order to rectify the mitochondrial abnormalities in the future generations, the scientists have brought a concept called as three-parent baby concept, which carries 23 chromosomes from original father, 23 chromosomes from original mother, and mitochondrial DNA from donor mother. Mitochondrial DNA from donor mother. This is to rectify the mitochondrial DNA problems in the future generations. Okay? Right. Over? Over? Right, next. Stem cells. What are stem cells? These stem cells are also called as base cells. The stem cells are also called as a base cells. These are undifferentiated primitive cells. These are undifferentiated primitive immature cells. So, undifferentiated primitive immature cells which have the capacity to develop into any kind of cells, which have the potential to develop into any kind of cells. So that means these are undifferentiated primitive immature cells which have the potential to differentiate into any kind of cells. So undifferentiated means unspecified, not specified. Okay, primitive, immature, not completely mature, have the potential to develop into any kind of cell. That means what? Suppose say this is some stem cell. You consider this is a stem cell, right? This is undifferentiated. Undifferentiated means what? It is not yet specified, and primitive immature cell. It has the potential to modify it into a skin cell. It can modify it into a skin cell. It can modify into a blood cell. It can modify into a cardiac cell, heart cell. It can modify into a liver cell. It can modify into a nerve cell. It can modify into a bone cell. Whatever it may be. Got it? It is an immature cell. It has the potentiality. It has the capacity to modify into any kind of cells. And these type of cells we called as stem cells. Stem cells are undifferentiated, primitive, immature cells which have the potential to develop into any kind of cell. So these are called as stem cells. Got it? And here the stem cells, based on its origin, the stem cells are broadly divided into, they may be embryonic stem cells, fetal stem cells, adult stem cells. So based on the origin, the stem cells have been classified into embryonic stem cells, fetal stem cells, adult stem cells. So embryonic stem cells. So the stem cells which originate from the embryo. So that means after fertilization, the zygote is formed here, and zygote undergoes multiplication, thereby it forms a group of cells. So 42 to 45 days of pregnancy you called as an embryo. So in this embryonic stage, up to embryonic stage, all cells are similar in their structure and function. And these type of cells we call as embryonic stem cells. Are you getting? So after pregnancy, up to 42 to 45 days of pregnancy you call as an embryo. In this embryonic stage, all the cells are similar in their structure and function. And these are called as embryonic stem cells. Then fetal stem cells. After embryonic stage, the fetus stage will be started. That is immature baby. That immature baby we call as fetus. Okay. And in adult stem cells, in our adult body, in adult organism. For example, bone marrow. Bone marrow is an example of adult stem cells. Bone marrow. So in this bone marrow, a tissue is present This is called as bone marrow tissue. From this bone marrow tissue, some type of cells are formed like RBC, WBC, platelets, and also limbus system cells. Limbus cells in our buccal cavity, in our oral cavity, on cheeks, in the interior of the cheeks, some cells are present. These are called limbus system cells. So simply you remember, based on the origin, the stem cells have been classified into embryonic stem cells, fetal stem cells, and adult stem cells. Embryonic stem cells means the stem cells which are present in the embryonic stage. Fetal stem cells means the stem cells which are present in the fetus stage after completion of embryonic stage. After 45 days of pregnancy, that you call as fetus. And then adult stem cells. So in adult organisms, that is in bone marrow and limbus cells, these are called adult stem cells. Got it? So this is based on its origin, based on the origin. Right? And next, based on the potentiality. So based on the potentiality, or based on the differentiation capacity, the stem cells are broadly classified into totipotent stem cells, pluripotent stem cells, multipotent stem cells. So based on the potentiality, the stem cells are of different types: totipotent, pluripotent, and multipotent stem cells. Totipotent. So totipotent stem cells. Totipotent stem cells means there is no restriction here. A totipotent stem cells can differentiate into any kind. Can differentiate into any kind of cell. Say for example, fertilized egg, that is zygote. Example, fertilized egg, or you can say zygote. So zygote is an example of totipotent stem cell. So what is totipotent stem cell? It can differentiate into any kind of cell. Say for example, embryonic cell. Embryonic cell is an example of totipotent cells. There is no restriction here. Okay. Next, pluripotent stem cells. So pluripotent. These are the descendants of the totipotent stem cells. Descendants of the totipotent stem cells. So these are the descendants of the totipotent stem cells. That means they have some limitations. They are not differentiated into any kind, but they have some limitations, some restrictions. They can differentiate into some kind of cells. Say for example, fetal stem cells. Fetal stem cells are called pluripotent stem cells. Pluripotent stem cells. And then multipotent stem cells. So these are descendants of pluripotent stem cells. These are the descendants of pluripotent stem cells. These are the descendants of pluripotent stem cells. Example, bone marrow. Bone marrow stem cells. So multipotent stem cells. These are the descendants of the pluripotent stem cells. Say for example, hematopoietic stem cells, which are present mainly in the bone marrow. These hematopoietic stem cells, they can modify into different kinds of blood cells like RBC, WBC. So they have more limitations. So actually totipotent means there is no limitation. It can modify into any kind of cell. Pluripotent means it is having it is a descendant of totipotent. That means it has having some limitations. It is having some limitations or restrictions. Multipotent means it is having much more limitations when compared with the pluripotent stem cells. Example, bone marrow stem cells. They can differentiate into only blood cells, only to some certain type of blood cells. So these are called as stem cells. Okay, the stem cells are broadly divided into embryonic stem cells, fetal stem cells, adult stem cells based on its origin. Based on the potentiality or differentiation capacity, they may modify into totipotent stem cells, pluripotent stem cells, and multipotent stem cells. Then what are the applications of these stem cells? So what are the applications of stem cells? So studies of human embryos of the stem cells can yield multiple complex processes, like physiology of the body can be studied. Say for example, we can produce the stem cells, we can produce the liver cells by using stem cells. Okay, we want to study the physiology of the liver, how the liver is functioning. No need to study in vivo condition. So we can study in the laboratory itself by producing the liver stem cells, by producing the liver cells from the stem cells. Okay, so that means the complex human process can be studied by using stem cells. The complex information about the complex events in the human in the development of a human body can be studied. And then so human cells, the stem cells are used to treat various types of diseases, to treat the diseases like Parkinson's, neurological problems, Parkinson's, Alzheimer's, certain types of cancers. The stem cells are used in the treatment of various types of neurological problems like Parkinson's disease, Alzheimer's disease, spinal cord injury, heart diseases, burns. All these can be treated by using stem cells. Next, you write down national guidelines for stem cell research. National guidelines for stem cell research. National guidelines for stem cell research have been jointly framed by the DBT, Department of Biotechnology, and ICMR. Guidelines for research on stem cells being formulated by Department of Biotechnology and Indian Council of Medical Research, ICMR. Right? Next, National Stem Cell Registry. National Stem Cell Registry. National Stem Cell Registry. It is a government managed database. It is a government managed database. Government managed database of unrelated bone marrow donors. Unrelated bone marrow donors. National Stem Cell Registry. It is a government managed database of unrelated bone marrow donors. Its main aim is to find Right? Its main aim is to find matching donors. Its main aim is to find matching donors for treating patients with blood cancers like leukemia, and lymphoma, thalassemia. So blood related diseases like blood cancers, like lymphoma, leukemia, thalassemia, sickle cell anemia, and hemophilia. So this is National Stem Cell Registry. So what is National Stem Cell Registry? It is a government managed database, okay, of unrelated blood donors. Its main objective or its main aim is to find the matching donors for the patients who are suffering with blood related problems like blood cancers like lymphoma, leukemia, thalassemia, sickle cell anemia, and hemophilia. These are all blood related diseases which can be treated only by blood transfusion. Okay, right. So next, Manav, Human Atlas Initiative. Next, another one is Manav. Human Atlas Initiative. Manav, it is a Human Atlas Initiative. It was launched by DBT, Department of Biotechnology. So it is a project to construct a comprehensive map of every tissue of the human body. It is a project, Manav, Human Atlas Initiative. It is a project to construct. A comprehensive map of every tissue of the human body. Every tissue of the human body. Human body. It seeks to capture human physiology. At the tissue level in natural and disease state. It seeks to capture human physiology at the tissue level in natural and disease conditions. Right? It seeks to capture human physiology at the tissue level in natural and disease conditions. So that means it is a comprehensive, it is a project to construct a comprehensive map of the human tissues or the total human body. That means, suppose if you consider the liver, it is an organ. Liver is made up of liver tissue. Okay, how these tissues are arranged, what type of tissue is there. Okay, in a similar manner, each and every organ can be studied, it is mapped. Thereby, we can better understand the information or we can better get the insights of this particular tissue, physiology, how the tissue is working, how the organ is working. If there is any abnormality occurs, then what is responsible for those abnormalities, that can be traced out easily. Okay, this is the Human Atlas Initiative that we call as a Manav. Okay, right. And next one is, and what is the next topic? What we left out? Gene therapy, gene editing. Okay. Then we put subheading gene therapy. Gene therapy. We'll have a small break, then we'll continue.